Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video I am going to talk about chemistry of enzymes, enzyme classifications, mechanism of enzyme actions, enzyme kinetics, inhibitions, activation and specificity. And I hope this video will be helpful to you. So I will start with enzyme chemistry. Enzymes are biological catalysts. It is well known. You know that speed up the rate of biochemical reactions. Most enzymes are three-dimensional globular proteins, tertiary and quaternary structure. Some special RNA species also act as enzymes and are called ribozymes. Structure of enzyme. The active site of an enzyme is the region that binds substrates, cofactors and prosthetic groups and contains residue that help to hold the substrates. Active sites generally occupy less than 5% of total surface area of enzyme. Active site has a specific shape due to tertiary structure of protein. A change in the shape of protein affects the shape of active site and function of the enzyme. Now friend, active site. Active site can be further divided into binding site and catalytic site. It chooses the substrate and bind it to active site. It performs the catalytic action of enzyme. Now cofactors. Cofactor is the non-protein molecule which carries out chemical reactions that cannot be performed by standard 20 amino acids. Cofactors are of two types: organic cofactors, inorganic cofactors. Cofactor inactive proteins, and binding site you can see active proteins. Cofactor binding activate the protein. Inorganic cofactor, these are the inorganic molecule required for the proper activity of enzyme. Example, enzyme carbonic anhydrase requires zinc 2+ for its activity. Hexokinase has cofactor Mg++, 2+. Organic cofactors, these are the organic molecules required for the proper activity of enzyme. Example, glycogen phosphorylase requires the small organic compound. Types of organic cofactors. Prosthetic groups, coenzyme. A prosthetic group is a tightly bound organic cofactor. Example, flavin, heme group and biotin. You can see heme group beta chain, iron, polypeptide molecule. A coenzyme is loosely bound organic cofactor. Example, NAD+. So an enzyme with its cofactor removed is designed as apoenzyme. The complete complex of protein with all necessary small organic molecules, metal ions and other components is termed as holoenzyme of holoproteins. Now friends, substrates. The reactant in biochemical reaction is termed as substrates. When a substrate binds to an enzyme, it forms an enzyme substrate complex. You can see here substrates joins enzyme. Sites of enzyme synthesis. Enzymes are synthesized by ribosomes which are attached to the rough endoplasmic reticulum. Information for the synthesis of enzyme is carried by DNA. Amino acids are bonded together to form a specific enzyme according to the DNA codes. So ribosome, nucleoplasmic reticulum. So intracellular and extracellular enzymes. Intracellular enzymes are synthesized and retained in the cell for the use of cell itself. They are found in the cytoplasm, nucleus, mitochondria and chloroplast. Example, oxidoreductase catalyzes biological oxidation. Enzyme involved in reductions in the mitochondria. Extracellular enzymes are synthesized in the cell but secreted from the cell to work externally. Example, digestive enzymes produced by the pancreas are not used by the cell in the pancreas but transported to the characteristics. Enzymes speed up the reaction by lowering the activation energy of the reaction. Their presence does not affect the nature and properties of end products. They are highly specific in their actions, that is, each enzyme can catalyze one kind of substance. A small amount of enzymes can accelerate chemical reactions. Enzymes are sensitive to change in pH, temperature and substrate concentrations. Turnover number is defined as the number of substrates molecules transformed per minute by one enzyme molecules. Catalase turnover number 6 into 106 per minute. Nomenclature of enzyme. An enzyme is named according to the name of substrate is catalysis. Some enzymes were named before a systematic way of naming enzyme was formed. Example, pepsine, trypsin and renine. By adding suffix AZ at the end of the name of the substrates, enzymes are named. Enzyme for catalyzing the hydrolysis is termed as hydrolase. Example, substrates, enzyme products. Lactose, lactase, glucose plus galactose. Maltose, maltase, glucose. Cellulose, cellulase, glucose. Lipid, lipase, glycerol plus fatty acid. Starch, amylase, maltose. Protein, protease, peptides plus polypeptide. Now, classification of enzyme. A systematic classification of enzyme has been developed by International Enzyme Commission. This classification is based on the type of reactions catalyzed by enzyme. There are 6 major classes. Each class is further divided into subclasses, subclasses and so on to describe the huge number of different enzyme catalyzed reactions. Classification of enzyme. Enzyme class, reaction type. Example, oxidoreductases, reduction oxidation, lactate dehydrogenase. Transferases, move chemical groups, hexokinase. So friend, hope this video will be helpful to you. If you like this video, then subscribe my channel and thanks for watching.